Hi, my name is Alyssa McQuillan. I'm a biochemistry and public health double major and a member of the iCons program on the biomedicine track here at UMass Amherst. I am currently pursuing my thesis in Elizabeth Vierling's lab in the biochemistry department. Here, we are looking specifically at small heat shock proteins, which are key chaperones that play a large role in stress tolerance. These proteins prevent the irreversible aggregation of proteins that have become misfolded and therefore non-functional. There's been a lot of research on the role of small heat shock proteins in human health. This is because when the protein quality control network is either overwhelmed with damaged proteins or is somehow defective, it often leads to various disease states. Understanding the mechanism of small heat shock proteins and their interactions with other chaperones has a wide range of implications, including fully recognizing the roles these proteins actually do play in cellular stress, as well as in disease processes. Using plants, in particular Arabidopsis thaliana, as a model organism, we can better look at these proteins and how exactly they are expressed under times of stress. With there being many different families of small heat shock proteins in plants, the focus of my project are the class 2 cytosolic small heat shock proteins, the chloroplast-targeted small heat shock proteins, and the mitochondrion-targeted small heat shock proteins. Not much is known about how a plant would handle stress if these proteins were not in place. So, to test for this, one would need to knock out these genes encoding the proteins and see how the plant survives. The first mutant will have both class 2 small heat shock proteins knocked out. These will be knocked out via the CRISPR-Cas9 mechanism. This is a unique technology that has enabled researchers to edit specific parts of the genome. It consists of 2 key molecules that are responsible for introducing the change in the DNA. The first molecule is an enzyme called Cas9. This enzyme cuts the double-stranded DNA at a specific location of interest in the genome so that bases can either be added or deleted. The other molecule is a piece of RNA called guide RNA. Each guide RNA is a pre-designed sequence that guides the Cas9 to the correct part of the genome that is being targeted. This is a way of ensuring that the DNA is being cut in the right place in the genome. The other mutants to be studied in this project would be a combination of knockouts. These mutants will be created by crossing plants carrying single or double gene knockouts in order to create plants that are triple knockouts for all chloroplasts and mitochondrion-targeted small heat shock proteins. Understanding how these mutants behave and handle different stresses will be very insightful to how organisms can survive without these proteins, if they can at all. With no small heat shock proteins being expressed, it will be interesting to see how these plants will tolerate any kind of heat stress. Ultimately, these mutants will provide us key insights into how important small heat shock proteins are in these plants and in life. Thank you.